I'm going to talk about the use of environmental sequencing to study microalgae. Particularly, I'm interested in this algae here that it's very easy to find to break any stony coral into pieces. So they are called endolytic or boarding algae, and the green algae Ostriobium, it's like a siphonous green one, it's the most commonly found one. So from an evolutionary perspective, why would anyone bother to study this algae? So if you look at the phylogenetic tree of all the siphonous green algae, you see that it's sitting here alone among a bunch of other seaweeds with more complex morphologies. And Ostriobium is just, this tiny little siphon, and it has just one family, one genus, and so far only 3 recognized species, although there is a study showing that there are at least 7 clades. But still, very little is known about them because they live hidden inside of a limestone, and because it's harder to define species, based on the morphology. But despite they look very simple, their ecological niche is very specialized. So they actively dig into limestone, and they live there, doing their photosynthesis under very low light conditions. So this sample here, for example, it was collected like 100 meters deep. So the coral living tissue was washed off, and you see that the skeleton is still full of boarding algae. So it would be nice to understand how they adapted to these conditions. But first we need to know how many species there are and whether they all have like the same ecophysiological requirements, right? The second reason to study this algae is because it adds to our understanding of microbial communities as a whole. So it has been suggested that corals can adapt to changing environmental conditions just by changing the composition of their microbiota. This was called the coral probiotic hypothesis, and it led to the development of this hologenomic theory of evolution that says that the actual unit undergoing natural selection is not the individual organism itself, so not the coral itself, but the genome of the coral plus the genome of all its associated microorganisms, right? So the whole genome. The problem is that we don't know enough about these communities, But nowadays, the most convenient way to study these communities is just to use environmental sequencing or metabarcoding. So with metabarcoding, we just get like a huge amount of sequences directly from environmental samples. So there is no need to isolate and culture the samples anymore. We just get a piece of skeleton. We do PCR, amplification. So what people normally use is the 16S for bacteria and the 18S for eukaryotes. And you submit to MySQL. And this is super nice, except that 16S or 18S, they are not really good barcodes to study boarding algae, because algae evolves very slowly when compared to bacteria, for example. So we need markers that have a better phylogenetic resolution here. So my main goal was to develop a protocol to study this endolytic algae using metabarcoding and assess the diversity of limestone algae, living coral skeletons. For this, we collected 160 samples in Western Australia and Papua New Guinea, and we decided to amplify the TUF-A gene, the chloroplast gene, because TUF-A has been suggested to be a good barcode to study algae. We also sequenced other markers. We also sequenced the 16S, 18S, and the 23S just to comparison, but I'm going to focus on the TUF-A today. And we submit for a few MySQL runs. So you get all this bunch of sequences, you do a quality control, and then we have to define operational taxonomic units. And they are based on a similarity threshold. We use it 98% because that's the closest we can get today to the species level. And then we built a maximum livelihood tree with these OTUs and other members of the chlorophytes. So the idea is to see where those OTUs fall in the tree of the chlorophyte. And then we explore the origins of endolytic nature and possible diversity patterns. So what I'm going to show today is what we have been seeing in terms of phylogenetic diversity, and a few thoughts about these diversity patterns. But the idea here is not actually testing these diversity patterns. The idea is just to show the directions of the research things that we're going to be doing in the near future. So the results. Using the TUF-A, we obtained 72 green algae OTUs, following the chlorophyte clade. From those, 52 match with the Ostriobium clades. So they are here, the O2s that we found are in red, and in black are the other chlorophyll known sequences. And this is just a zoom in the Ostriobium clade. Unfortunately, we only have like 2 sequences for the known Ostriobium species. We don't have the sequences for all the described species. But it's clear that there are far more species than the 3 ones that are described so far. When we use the aging acid gene fragment, we obtain only 20 O2s, and they are mainly dispersed through the tree. So they're not like, most of them don't actually match with Ostriobium. Sorry. I have to drink water because I got a bit of a cold, and then my supervisor told me that could take over if I start coughing too bad. I've got a bit more convincing, right? So, we also found several O2s that are more distantly related to Ostriobium. For example, this clade here is like very distantly related to all the other algae that we know. So these are 2 different genera. So this could be an entire new family of like exclusively endolytic algae. And some other O2s actually match with no seaweeds. So this is interesting because the seaweeds, we don't really know like about their life cycle so much. So they could be just endolytic stages of their life cycle that is not described yet. Then one interesting question is to see like where does the endolytic nature came from, and we see that they come from everywhere. So we mapped the ancestral states in the phylogenetic tree, so everything that we observed like an endolytic O2 or that was previously described is marked in black. And we see that the endolytic nature appeared several times in the phylogenetic history of the chlorophyll. Ostriobium is here. And Ostriobium is very old, so it appeared like 500 million years ago. And it's actually like 300 million years older than hard corals. But this matches with the appearance of the first metazoa reefs. So boarding algae started to board into limestone way before its association with corals. So we saw that there are several lineages, and then we asked, but do these lineages have different ecological niches? So we mapped the presence or absence of these OTUs on samples collected across a depth gradient, so from 0 to 50, and in different coral genera. And we didn't see any pattern. It's very frustrating. But not completely unexpected, because it has been shown for social study that what actually changes with environmental agents is not the presence or absence. They are generally everywhere. What changes is their abundance. So this just raises more questions, Does this lack of a pattern is because they also share the same ecological niche, or do their abundances differ? And how does it relate to the coral holobiomes, especially? So why does it matter for the coral? So boarding algae are one of the main agents of reef bioerosion. Because they board in the coral, they kind of weaken the skeleton, as you can see in this picture here. And it has been shown that with oceanification, climate warming, the bioerosion mediated by this algae increases even more. On the other hand, it has also been shown that endolytic algae can provide an alternative source of energy when corals bleach. So our next step now is to understand how does the endolytic community, or how does these different lineages relate to environmental factors, and how do they relate to corals' health and disease. There is still some drawbacks on this sort of work, especially due to PCR bias, so when you do PCR, some DNA is amplified more than others. The size of the gene fragment, so my seq today provides one of the longest read lengths, but this is still like 600 base pairs. This is still not really big to construct phylogenies out of that. We still need better methods to define OTUs that allow us to get to the species level. Meanwhile, we're just using similarity thresholds. And reference data sets, because they have for 16S and 18S, so we can match the things that we're finding with sequences that are already known. So to conclude, I think this is the proof of concept, just that environmental sequencing can be used to study microalgae and probably many other microcariotes. We saw very high diversity of evolutic algae, especially within the Australian plate, but also with a lot of undescribed lineages. And the next step is to describe the relationship between the community composition and environmental factors. So in the long run, we expect to combine the information that we obtain about the evolutic communities with the communities living in the coral tissue itself or the coral mucus, it's more commonly done, and then relating how all those communities react to environmental factors and how this all relates to coral's health and disease. We may understand a little bit more of how the evolution works. And that's it. Thanks for people who helped in the lab and the field and founding. And thank you for staying to the last minute of the presentation.